So today the internet is a little bit troubling, but we'll try to finish this lesson. Okay, good evening, everyone. We will talk about insulin, not insulin as such, but we will talk about actually about a research paper that we are supposed to read as a part of our fourth unit. Okay, and then we'll have NGS, we will have some the piggyback vectors, one part, and we'll also have CRISPR-Cas. Those are the topics that will be still pending. Okay. So in today's class, we will talk about mostly about diabetes. Okay. Let me go and discuss diabetes first. What is diabetes? There are two things: diabetes mellitus and diabetes insipidus. Insipidus is about lack of vasopressin and reabsorption of water is the problem. Here, diabetes mellitus is characterized by high blood glucose. Blood glucose, GLC, is the three-letter code for glucose. It's not GLU. GLU is for glutamic acid, three-letter amino acid. Okay, just remember that. So there are two types of diabetes, actually three or multiple are also there, but we will talk about type 1 diabetes and type 2 diabetes. Diabetes is lack of insulin function. Okay, I'm not saying insulin per se, I'm just saying insulin function. What I mean by insulin function is that the function of insulin is to ensure that glucose is taken up by the cells. How does it do that? Normally, the cells, the skeletal muscle cells or liver, they have vesicles with GLUT4, the facilitative or passive way of glucose export. These importers, you can say GLUT transporters, GLUT4, they are present on, one second, sorry. Yes, they are present as vesicles in the cytoplasm, cytoplasmic vesicle, you would call it. So they are not actually transporting or anything because they are all in the cytoplasm at this stage. But when insulin binds to the insulin receptor, IR is insulin receptor, it activates another molecule called as insulin receptor substrate 1 that results in the production of PI3K, phosphatidyl inositol 3 kinase, then the production of PDK and activation of PKB, and eventually these vesicles are made to fuse with the cell membrane. And so then now GLUT4 is on the cell surface, and then it will take up the glucose from the outside, and it will put it into the cytoplasm. That's how the regulation of insulin, the insulin will cause the uptake of glucose. But what may happen is there are two types of diabetes, we said. Inability to produce insulin. That means insulin is not present at all. That is one problem. That is type 1 diabetes. Type 2 diabetes is the responses, the target cell is not responding to the insulin that is present. So it is not taking up glucose, because of which the glucose in the blood increases. That is hyperglycemia. Mostly it is because of the input of food is too high calorie rich diet, which digests very fast, which shoots up blood glucose very high, very fast. That is called as caloric glycemic index. And there is no output, zero output in terms of expenditure of that, output in terms of work, physical work. Okay, so these two are biggest reasons for diabetes. Say we are eating food because we are hungry. We are eating food because others are eating food. We are hungry. We are eating because we are bored. We are eating because there is food. That much of food is available to us. It's constantly available. And we are shooting our blood with a lot of glucose all the time. And the pancreas is producing insulin all the time. but we are not spending it because of our sedentary lifestyle. So the cells are becoming used to the amount of insulin that is secreted into the blood, and it develops a kind of insulin resistance, and because of which they don't respond to insulin as much. And there are also other causes such as mutations which might in some of these proteins, which may result in the resistance to insulin. So there are uh, some people sometimes have low blood sugar, and some people sometimes we have high blood sugar. Some of the symptoms is we are always dry, we urinate, frequent urination is there, and bedwetting is also a possibility. We are drowsy, and sometimes we drink a lot of water as well, because there is too much glucose in our blood, right? So the kidney will try to pump it out, and along with that there will be a lot of urine water loss as well. Because we are losing a lot of water, we end up drinking a lot more water. So that is how we usually some of the conditions result because of hyperglycemia. So having high concentration of blood glucose is dangerous to multiple tissues. Mostly it can be nephropathy. The kidneys are affected, they are overworking, and then they have a lot of problems. Cardiomyopathy is one of the other biggest problem, and retinopathy. If you have any old people around you, you might actually have noticed that with those with diabetes mellitus, they have diabetic retinopathy. It also affects the brain and teeth. So you can see that just blood glucose, having high concentrations in the blood, is affecting so many different tissues, and it is not so nice. So if you look at diabetic retinopathy, what happens is there are two problems: hemorrhages, that is blockage or splitting of the blood vessel and leaking the blood into this, and microaneurysms. There is bulb-like structures occurring in the blood vessels, because of which the retina is around here, and if it is masked by these hemorrhages or aneurysms, the visibility will be gone, some parts. This is how it usually looks. A normal person can see as clear as this, and a person with diabetic retinopathy may miss spots where the person cannot see because they are obstructed by hemorrhages or aneurysms. There is another problem, is that in the at least in feet and other peripheral blood circulatory system, you can see there can be damages. So they call it as diabetic foot as well, where the blood vessels become narrow, right? And because of that, there is not enough supply of blood. And if blood is not supplied, it's not in sufficient quantity, the nutrition is missing and oxygen is missing, and also accessibility to the immune system is missing, right? There are multiple outcomes because of the narrowed down blood vessels. So when something like that happens and there is a obstruction immediately, and then there will not be any more supply of blood to the lower part, and that can cause serious injuries, especially the feet. Under the feet also, people, because we put all our weight onto our feet, this is a big spot where area which is taking a lot of pressure of our body, and then they could be affected and they could die. The cells there may undergo necrosis. In extreme further sudden cases, you might have all of this bluish tissue is actually starting to die, necrosis. Cells are dying and spilling over everything else. There is no option but to amputate this region, because there is no way of healing in this part. And it can result in removal of feet, or it depends on until where the injury is. Say, for example, this is where the blood clot has happened. There is no point in cutting till here, because upon amputation, healing should have happened. For healing to happen, there should be good supply of blood. So usually in this case like this, they may amputate until here. It depends on the scenario. Okay, I hope we have generated some motivation about why we should be learning about it, and working towards bettering these kinds of complications that happen with many people. So insulin as such has multiple functions. The first of it is apart from glycogen uptake, it also has, so say this is the cell membrane. The glucose that is present here is made to be taken up by insulin, and this insulin should be made into glycogen storage in the form of glycogen. So whenever this skeletal muscle cell or liver is required, they can break down the glycogen to glucose and use it up, or secrete out to the other tissues which might require. Okay. Insulin is one of the smallest protein. When I say smallest protein, what I mean is because it kind of has a quaternary structure. What is a quaternary structure? The sequence of amino acids is the primary structure. These folding into beta sheets or helices is secondary structure, and how they are organized among them is the tertiary structure, and how several subunits like this come together and function as one enzyme or protein is the quaternary structure. Insulin has a kind of quaternary structure. Of course, when we see the processing of insulin, you might have already learned this, but that's why we will just have a quick review of what it is. It consists of 51 amino acids. There are 2 chains. That's why I'm saying it kind of has quaternary structures. 2 chains bound by 2 disulfide bonds. And one chain is 30 amino acids, and the other one is 21 amino acids. It is produced by the beta cells, okay, beta cells, and it is produced as proinsulin, which is a single polypeptide. And later, this part from here to here, the gray one, is the free chain C is removed. And what is left then is the mature insulin. So it is produced as pre-pro insulin. When it is produced, I will just discuss it here. Say, for example, this is the cytoplasm. So this is the ribosome here that is producing it is translating the mRNA of insulin. Using the signal peptide, the insulin is prepared and secreted into the rough endoplasmic reticulum, RER. And there, at that stage, when it contains the signal peptide, we call it as pre-pro insulin. Upon that, it is processed. The signal peptide is cleaved away here, and what is left is then pro-insulin. This pro-insulin is further modified when it is in the secretory vesicle. There it loses the chain C, and now what we have is chain A and chain B attached together with 2 disulfide bonds. Okay. So what these enzymes, the insulin does, insulin kind of regulates many more things actually. It is about maintaining the blood glucose, and glucose is one of the primary energy sources in majority of the organisms. And somehow we have to produce glucose, especially say tissues like brain. They need glucose all the time, glucose and oxygen. They are very intolerant to lack of oxygen or glucose. And that is also, that means glucose is playing a central role in metabolism and conversion of gluconeogenesis. I hope you know what is gluconeogenesis. When we do not have glucose in the body, then amino acids could be broken down into amino acids. The proteins could be broken down into amino acids, and those amino acids, from those amino acids, we can synthesize, the cells synthesize glucose, that is enhancing gluconeogenesis, and which can be secreted out. And this happens when we are in prolonged starvation situations. So we know about diabetes, and we discussed about two types of diabetes: diabetes 1, type 1, and type 2. Type 1 is lack of insulin. That means somehow the cells are not, the beta cells that produce insulin are, there is insufficiency of insulin. Type 2 is resistance to insulin. There is not much we can do about type 2 diabetes, in the sense that if it is because of our lifestyle, the sedentary lifestyle, we should try to keep ourselves very active all the time. In type 1 diabetes, which could be caused because of autoimmune disease where the beta cells are killed away, because of which the lack of beta cells, there is lack of insulin, and that's why blood glucose is high. So we can try to provide solutions for it. Because in type 1 diabetes, we say it is lack of insulin, we can use cloning. That is where rDNA comes in. We can construct recombinant DNA and produce insulin. So in previous olden days, they used to take the pancreas of pigs from the slaughterhouses, and from them they used to collect insulin. Just to get a gram of insulin, there used to be so many pancreas required. And now, because of genetic engineering or recombinant DNA technology, we use E. coli to produce the insulin that we want, right? Like, that is the course that we are actually doing. So in a typical exam question, we could ask you, how would you, if you were in the situation where you have to make insulin, then how would you do that? So there are multiple issues about producing insulin, because insulin, we are saying, is not a single polypeptide. It is rather two different chains attached by disulfide bonds. How would we produce it? The simplest, the logical conclusion would be that if you are producing it in E. coli anyway, E. coli does not have all the enzymes required to produce an final insulin product. I'll just go with this so that here I could probably be able to explain. If it were a single gene which will produce one mRNA, and then that will form one protein, it's easy and straightforward. But that's not how insulin is. Insulin is like this. How can we then make insulin like this? We should have made chain A first, chain B first, next, or in two different cells, and then somehow be able to make disulfide bonds between them, right? So in this protocol, I'm not going to discuss whatever I'm going to discuss now is not complete details. I would want you to work on it and find out the literature about how is the final insulin made. I will only go through a few things. So as I said, we can clone a gene in one plasmid, the chain B, or open reading frame for chain B in one, and open reading frame for another one in another plasmid. But then they are made as translational fusions, beta galactosidase. Why should they make as translational fusions? Why not directly? Usually, when we are producing a foreign peptide or protein in E. coli, they are likely to be degraded by proteases. This is especially true if they are small. Small in the sense, one we said there's 21 amino acids and the other chain is about 30 amino acids. So they are unlikely to be stable within the cell. Then we wouldn't get enough quantity. So to make them stable, we need to make them as translational fusions. So assume this is chain A and beta galactosidase. They made a translational fusion. So when it is produced, it produces a protein like this, something like that. So this part you can assume as beta galactosidase. This is the part chain A. What is shown here, the black one is the beta galactosidase in both the cases, and the remaining is the either chain A or chain B. So we can then purify the proteins that are produced out, and then we should be able to cleave them in here. How can we cleave them? If you want to cleave them, we should have done something else. We should have put a specific sequence or motif that can be recognized by a protease. And so the protease is motif specific, so it will bind here and it will cleave this place. And then using chromatography, we can separate the alpha A chain and B chains, and then we can put them together, and using chemicals, we can make the disulfide bonds. So here is what you are supposed to read and find out from the literature. And when the question comes in the exam, you should be able to answer. Is how do they do it actually in the paper, in research? You have to delve into more about why, how this chemical reaction is made. In genetic engineering or rDNA, it is never complete until here. Just construction of recombinant DNA is not the outcome of it. The outcome of it is about the product. What product have you made, right? So each say because this is insulin is a common thing that we all must have been studying. So I want you to have the specific details of how these things have happened. I didn't tell you about the protease and the cleavage site. There could be other mechanisms also, how they are cleaved. I did not tell you how the separation is made. I did not tell you how the purification is made. I did not tell you the specific details. I want you to find out those specific details in each of those steps, and also about the how disulfide bonds are made. Okay, please note it down and write it. You don't have to submit anywhere. All I need is when in the exam, you should be able to write specific details, not superficial. Okay, so I think I already discussed this, and we will move ahead. And now, instead of using pig insulin for treating diabetes 1, we are now able to make, because there are differences between the insulin of pigs, although it might be effective enough in humans, there are differences between insulin of pig with that of humans. The problem is, if this pig insulin could activate the immune system and then be degraded continuously every time we are injecting pig insulin. So the best possible way is to have human insulin, right? Because then the insulin will not be cleaved or degraded by the immune system, because it's a natural one that is present in the humans. So now with recombinant DNA technology, we are able to do those stuff. Yes, the actual paper that is in your syllabus is about this one. Whatever I gave you, I spoke of till now, is actually the introduction of what I want to speak. It is about, the title reads Engineered Enteroendocrine Cells Secrete Insulin in Response to Glucose and Reverse Hyperglycemia in Diabetic Mice. So their model organism is mice, and this is based on kind of gene therapy, and they are proposing it as a mechanism or some flowchart um, for type 1 diabetes, where there is lack of insulin production. Let's still have some more details before going into what they have done, and a little bit we'll see. Insulin is produced by beta cells, and they could be destroyed by multiple things. So often it is about viruses because of excess body weight, and there could be there is also gestational diabetes that I haven't discussed. Stress, microbial toxins. There are numerous environmental chemicals that also affect the beta cells. So one of the things that happens is, say, sometimes because of the viruses, infections, there could be genetic susceptibility. In some people, the beta cells are more prone to be attacked by the immune system of their own body. Sometimes it could be triggered by environmental factors, including bacteria, nutrition, chemicals, and so on. So what may happen is some of the cells may be killed by the immune cells, and then the immune cells will now present more proteins on their cell surface, because of which now B cells from the immune system, antibody producing ones, they might be triggered towards the antigens that are presented, B cell antigens. That means we are going to have more autoantibodies against the beta cells. And eventually we will have so many autoantibodies that the majority of the beta cells are killed away. So this is where that will result in hyperglycemia. So why we are looking at this is, we had beta cells, they are killed by antibodies or the immune system. If we look it into stem cell based methods, what is the ideal way is to put, because beta cells are missing, insulin missing, so put in beta cells, they will produce insulin. But then the antibodies may attack and kill the newly implanted beta cells also. Then what is the solution then, right? So people have identified some particular antigens that actually trigger the immune response, and they're trying to work on that. We have to look more into those details too. But I hope you understood the point. We have beta cell death is because of immune system, and if we put more new beta cells that can produce insulin, they could also be attacked by the antibodies. So let's see, in the pancreatic islets, there are multiple cells. You have beta cells, alpha cells, delta cells, and this is how a normal islet would look like. Whereas in the type 1 diabetes, because the beta cells are destroyed, you will have them very few in number, or non-functional and dying. So the solution is beta cell replacement. That's the concept that I was speaking about in a few slides, the last before slide. So one of it is to try to regenerate these beta cells. If there are somehow using stem cells, if we can trigger them to develop, then probably they would be able to solve the issues. But then we have a lot of problems with that also. We can take from allotransplantation, which means we could take from other people and provide. but then the risk of infection is very high. And in the long term, it is not going to be very viable. And xenogenic is trying to take from some other species like thing, and they might have different difficulties with insulin um, compatibility and also immunocompatibility. So because we are saying that beta cells are attacked by immune system, we could try to have some ways of how immunoisolation, that means trying to put them into with membranes consisting of immuno resistant ones, meaning that the antibodies or the immune cells may not be able to traverse through this membrane. So if we put beta cells there, and they might probably produce insulin, but that's still not a very solid solution. It is a kind of solution, but there will be a lot of issues if it is not allowing immune system to go here. That means this, if there is any kind of damage here, it cannot be cleaned up by the immune system. Secondly, if there is problem with inflow and outflow, the requirement of the niche conditions, the growth factors, the supply of everything is not in proper way. So we need to find a different solution and how to do it. Whatever solution we come up with should be able to produce insulin and process it, right? Should have the gene, and gene should be expressed, and that whatever is produced should be processed to form mature or completely formed insulin. Second thing is it should be secreted in time. I hope you have seen some people injecting insulin on a daily basis at regular times, right? When we eat food, the blood glucose increases, and that is when we are supposed to give the take in insulin, so that the blood glucose will come down. The timing of insulin secretion should be quite normal. We will discuss a little bit about it today in a while. And the third point that we have to keep in mind is that we should somehow be able to protect the cells that we are putting in, right? And which should be the quantity of the number of cells that we put in should be fine, should be enough. And the timing, as I said, they should be here, the cells should be able to secrete insulin, and they should be able to secrete insulin in sufficient quantities in this sufficient at the appropriate time, right? Anytime a question is given to you and you are asked to provide a solution, and in that case you have to provide solutions thinking or trying to break down what are all the challenges that you might have, not just going into the from here to here producing the answers, unless you know what are the challenges that you have to overcome. Then only then, if you know them, then you can provide a sufficient solution, and that is actually the best idea possible at that time with the available resources. First thing, let's look into some biology today. And food, when we eat food, the food is, we chew food, it start digesting in our salivary amylase, and with insulin in the intestine, the food is taken up, which usually consists of glucose, fatty acids, and so on. And they will reach the blood, through blood they will reach the pancreas, and in pancreas the beta cells are triggered to secrete insulin, and that insulin goes through the endocrine system, it goes through the blood to all parts and stimulate the target cells to take up that glucose. The target cells are typically liver and the skeletal muscle cells. So these are, liver is kind of storage for a lot of glycogen in the form of glycogen. Skeletal muscle, because we will need to work, and they also store in the glucose as glycogen. So what happens between the beta cell and the target tissue, muscle. So assume that this is somewhere, this is the intestine, and the glucose is taken up into the blood. So it reached the beta cell in the pancreas, and there is the GLUT2, glucose transporters 2, are present, which will take up the glucose, and glucose upon glycolysis will generate ATP. So when there is an increase in ATP to ADP ratio, it is going to activate ATP sensitive potassium channel, and that is going to cause depolarization of the membrane. Membrane depolarization occurs, and that membrane depolarization, that the voltage that is generated is um, triggers a calcium channel that is dependent as voltage dependent or voltage gated calcium channel, because of which the calcium that is present in the extracellular side will now go into the intracellular side. I mean, there is increase in calcium ion concentration within the beta cell, and this increase will allow the fusion of these vesicles, insulin containing vesicles or granules, with the cell membrane, thereby releasing the insulin into the blood. Okay, until then insulin is produced already, processed, and put as vesicles or granules within the cell. Okay, it is not secreted into the blood system, but because of the increase in blood glucose, there is a trigger, and now insulin is produced, secreted into the blood, and this insulin is circulated, and it would bind to the insulin receptor, which would get phosphorylation, and then there are several things that will happen, IRS, PKB, PDK, and so on, and then they will allow the fusion of cytoplasmic vesicles containing GLUT4. This is GLUT4 here, and in beta cell, it is a different glucose transporter, that is GLUT2. So the vesicles will fuse with the cell membrane, and now they will take up glucose. Okay, so that is the integrated system between what is ingested into the intestine, what into the mouth, stomach, and then into intestine, and how the whatever the food that we ate becomes a part of the target cells, and that is done by the insulin. So we want insulin secretion only when there is glucose increase in the blood glucose increase. So I'll skip this because I think we have already discussed this. We don't need all the details at this point, but still I think we have considerable things we have discussed. So the point that we are trying to make here is we cannot use beta cell because it is under attack by the auto antibodies. So we have to find some other way, some other cell type that can still produce insulin, that can still be able to secrete insulin, that should be able to time the insulin secretion based on food intake, right? These are our goals then. So if we look at the stomach or the large intestine, the small intestine, we can find several types of cells. Out of them, some of the cells are called as enteroendocrine cells. They secrete several hormonal products that are timed based on the intake of food. So the paper that we are discussing, they made a choice and they chose enteroendocrine cells because they can time their activity or secretion. The timing is appropriate. Timing is after ingestion of food or when the blood glucose is increased. They have many other functions in the body, as I said, production of GIP and others. So what these people, the others, have proposed or tried to do is they made they they have chosen the cell that is going to be enteroendocrine cells. So they're producing, they made a recombinant plasmid like this, which is called as GIP. We'll have 1 second. I thought I had a slide about those things. We will see if I have to repeat. Ampicillin resistance gene, that is for making all the manipulations in E. coli. All genetic manipulations are done in E. coli. So we are going to use ampicillin resistance gene. Of course, when we say that, we will also have origin of replication for E. coli as well. Then we are going to, they have neomycin resistance gene. This is for selection of cells, like we have discussed in the last class, selection for the animal cells. Animal cells that have obtained this plasmid from those that did not obtain it. Then they have made a the recombinant, just for this part, they have made something like this. They put a promoter from cytomegalovirus and they put the gene for insulin. And they also have a promoter for GIP, because GIP is secreted and produced in enteroendocrine. If they put the GIP, okay, this is the important thing, promoter for GIP, it would be produced within enteroendocrine cells. As you may understand, insulin gene is present in all the cells, right? In all the cells it is present, but only beta cells will produce insulin, which means that only insulin is expressed only in the beta cells, and then the machinery will process it and produce insulin. In all the other cells, although gene is present, they are not, because it is not produced, because it is repressed. It is silenced within the region. So now we want, our goal is to have insulin produced in enteroendocrine cells. And in enteroendocrine cells, GIP is produced. So what we can then do is we put GIP promoter, because GIP promoter is active in enteroendocrine cells, and make a transcriptional fusion. Here we are not putting GIP, but we are putting insulin gene. So because this promoter is active in enteroendocrine cells, we will have mRNA produced and then the protein produced. There are problems again. The problem is the insulin that is produced should have been actually, it should be what you call processed. So enteroendocrine cells, I'm missing the slide. Maybe in the next class I will try to get it. I'll see if I have. It should be processed. And so these cells also have the processing machinery. What I want you to remember here is that what we are discussing as a biology is of humans. And what we have here, the model organism, because people cannot do experiments directly on humans, they try it in smaller mammals, which are close enough but not identical. So these people have tried in mice, and mice, the insulin is a little bit different. I think they have two forms of insulin. Anyway, right now we are trying to understand the concept of what they have done and how they would have characterized. So this is, they had the GIP promoter, and here is the insulin gene, and then they have this region for intron sequence, um, and then polyadenylation signal, PA. I hope you remember that from our previous discussion as well. Okay. I'll just discuss this one last and then I will stop. Just I'm giving a very simple uh, discussion. We will repeat in the next class maybe. So this is what GINS, and this is in the intestine. These are STC cells, and this is another MIN cells. You can see in the legend if there are those things. What they have done is to check if which all genes are expressed in these different cells. Okay, I will repeat. It is a reverse transcriptase PCR. I hope you remember what is reverse transcriptase PCR. It is not real-time PCR. Real-time PCR is different from reverse transcriptase polymerase PCR, which means they may have similarities, but there are differences. RT-PCR is more of a qualitative one. We can determine if something is expressed or not. Real-time PCR is qualitative as well as quantitative. It can tell us how much of transcript is present, how much of the template is present. In this paper, in this work, they have used reverse transcriptase. So it is more of qualitative one. It will say whether a particular gene or mRNA for a particular gene is present or not. So here we have different, on this scale, on this way, there are different cell types, and here different mRNA, different genes that they have tested for, or mRNAs that they tested for. GADP, GAPDH, this glyceraldehyde 3-phosphate dehydrogenase, which is kind of controlled, present in all the, expressed in all the cells, is expressed in all. You can see these bands. We cannot make much out of what is high in concentration and what is low in concentration. That is one of the problems here. And again, in real-time PCR, we usually measure in terms of chord curves. I hope you remember those things that we discussed earlier during this course. GLUT2 is, we discussed that it's a glucose transporter that is present in beta cell, right? We can see it highly expressed in Min6, and there is very little or none. There is, but. And then they have tried for several of it, and they have tried also for this insulin that they have put in. It is not present in any of them, but except in the cells, they have gastrointestinal enteroendocrine cells that they have put in the plasmid with. And that has produced sufficient amount of this insulin that is put in. And this is the mouse insulin that is being produced by this. This is a control here. Okay. We'll try to discuss this again in the next class, but I hope you can, the goal of such papers is your ability to troubleshoot. You take a problem, you take the problem, and you dissect it into multiple sub-problems, because each of it goes in steps. Each of it is a challenge. And then how do we address each of them, and what are all the ideas that are available? And then you come up with the best possible idea. And that is how we go about ideation. And we have to always take such broad and, in terms of thinking, a luxurious idea. We cannot just go by, I'll only think this much, or I'll only consider this much. That's not how things happen. We have to think a lot more details. Even this is, in this paper, they show that they have successfully reversed or allowed the uptake of glucose to a great extent, but still there will be limitations, and that's for the future work to be carried out. Okay, I'll stop here. And I think tonight or so I will give the answer, the